Has she cured cancer? Not yet, but a UVM undergrad from Swanton, Vermont did just win one of the nation's most prestigious science prizes. We're learning about her path and her love affair with lab work today on Across the Fence. Hello, Vermont. 40 years ago, when the Goldwater Scholarship was established, the mission was clear. Identify the brightest young talent emerging in the natural sciences, mathematics, and engineering, and support and nurture their careers as they became leaders and innovators in their fields. Today, the geopolitical imperative behind the Goldwater Scholarship may have changed, but the scholarship itself hasn't. It still acknowledges the best and brightest undergrads in the country, paving their way into prestigious PhD programs, fellowships, and other amazing scholarship opportunities. Over the years, 15 UVM students have been named Goldwater Scholars. The 15th joins me today. Say hello to Madison Guyette, an undergraduate majoring in molecular genetics at UVM. She works in the Chatterjee Lab, part of the UVM Cancer Center, studying factors driving cancer onset and how to make treatment therapies more effective. Madison Guyette, thank you so much for joining me.
Yeah, thank you for having me.
It's wonderful to have you here. Although I'm going to, full disclosure, I will probably not necessarily be able to follow all of the science and all of the science. So I'm going to let her be the scientist and I'm just going to ask the questions today, okay? So tell us about your work. What have you been studying?
Yeah, so in the Chatterjee Lab, we focus on something called the translation synthesis pathway, or the TLS pathway. And what TLS is basically a DNA damage bypass pathway. So every day your cells are encountering lots of damage, whether it be from what you're eating or when you step outside and you get UV damage. So your cell needs a way to repair that damage, otherwise it will die. And TLS is a bypass mechanism. So what that means is it's kind of able to bypass DNA damage. Your cells have a lot of repair pathways in place, but sometimes if the damage is too extensive, they're not actually able to act on it. So TLS comes in and it's actually a low fidelity mechanism, which means that it'll insert random nucleotides at the site of damage to allow the cell to keep replicating. And we study that in the context of kind of cancer therapy. So TLS is implicated in cancer in kind of two ways. One, like I said, it's a low fidelity mechanism, which means that it has the capability to introduce mutations into the DNA. And if that disrupts some of the kind of checks and balances that your cell has in place, it can actually contribute to cancer formation.
Okay.
The second way, which we kind of focus a lot more on in the lab, is cancer resistance. So the main therapies that we have in place for cancer treatment right now are chemotherapy and radiation therapy. And kind of the aim of that is to induce enough DNA damage into your cells so that the cancer will die. But if you have TLS acting, your cells can actually withstand a lot more DNA damage and they can kind of bypass these therapies, contributing to cancer resistance. So we focus on this one TLS protein called Rev1, which actually kind of functions as a scaffolding protein, which means that it recruits other TLS proteins to the sites of damage in order to make this process happen. So we study how Rev1 functions and kind of how inhibiting this protein can be exploited for better cancer therapies.
This is great. See, this is why she is the future of cancer research. Okay, so my basic understanding is so you're understanding the mechanisms in the cell, in part because you want to, there's a possibility of understanding how to make cancer therapies more effective?
Yeah, so the idea is kind of with these joint therapy, like a two-step mechanism. So one is you use your regular chemo or your regular radiation therapy, and you do this in combination with some translation synthesis inhibition. And the idea is that because you're eliminating a mechanism where the cell can tolerate that extensive damage, your therapy will work more effectively.
And this is something that we want. We definitely want more effective cancer therapies. Okay, so before you found Dr. Chatterjee's lab, and we're going to talk about more about that in a minute, was cancer research personal to you?
I think with everyone, everybody knows someone personally that has either had or has cancer. It's a really big problem, especially in our country right now. Incidence rates are really skyrocketing. For me, both sides of my family have cancer or have had cancer. Both sets of my grandparents and my dad especially. So there is a little bit of a personal angle there.
Yeah, so that was a draw into the lab for you, that you're like, this is something actually where I want to be a part of the solution in the
Yes,
that's
definitely.
that's a very, very simplistic way of putting it, but yeah. Okay, well, in the middle of summer, UVM is a bit of a ghost town, except for students like Madison working away in the lab. I went over to the UVM Cancer Center to see what a day in the life was like. Let's take a peek.
And is it fine that that's loud? Okay. I think something that I really love about being a researcher is that there isn't really a typical day in the life of someone that's working in a lab. It is very variable depending on where you are in your projects and your experiments. So some days I might come into lab and I might spend the entire day in the wet lab space performing experiments, and other days I might be reading, I might be writing, I might be working with my mentors to kind of figure out the next steps of my experiments. I come from a school that didn't really have a lot of opportunities for science, and especially during COVID, the AP biology class that I was taking, we weren't really allowed to do any in-person experiments. So I didn't really know what research was at all. I remember coming to UVM for a tour and people asking me if I was interested in research, because UVM is a really big research institute and I had no idea what they were even talking about. I actually had never held like a pipette before. I spent a lot of time shadowing techniques in the lab because I wasn't familiar with anything and I needed to get kind of a lay of what science looked like. And then it wasn't until this past summer I got funding, so I was able to spend a lot more time here, and I was able to work really closely with another undergraduate in the lab, and he kind of let me do my own experiments along with him, so we were doing the same sort of things in parallel, and just being able to have ownership over doing something, that's when my love like really, really sparked for it. And then I was here all the time after that. So seeing the amount of growth that I've been able to have over the last couple of years has been really amazing.
Okay. The idea that you didn't even know what research was, that's incredibly astonishing. On the other hand, you had been taking AP Bio, so all of the raw materials, I would say, were there, potential was there. But talk a little bit about what it was like coming to UVM as a first-generation college student, because you were also the first person in your family to go to college.
Yeah, so I'd say the college transition for everyone is a little bit daunting. For me especially, the first couple of weeks at UVM, I definitely felt like I was a little bit behind my peers, especially in my major. I think a lot of people have had family members who went to college and were kind of able to guide them in that process and be able to kind of inform them about what opportunities their degree actually holds, versus I was kind of going in there blind. But I will say I got really lucky in that the microbiology and molecular genetics department, which I'm a part of, their faculty was truly really amazing. We spent a lot of time first semester taking some intro courses where they made sure that everybody was really at the same baseline. And I feel like I formed very good relationships with all of them, to the point where they kind of filled that gap where I couldn't necessarily ask my parents for advice, but I was able to go to them, and any question or concern or worry that I had, they were there to answer it for me.
Well, and now you're a Goldwater scholar, and I know that you said that in part it was because the lab and Dr. Chatterjee got so behind you. Talk a little bit about what the process was like winning, like applying for and winning the scholarship.
Yeah, so Dr. Chatterjee was a huge, huge part of it. I think it was our first or second meeting where she had mentioned, oh, I want you to apply for this scholarship 3 years down the road. So that was really amazing, seeing that she had my best interests in her mind at all times. And so one really huge, obviously the Goldwater scholarship is a really competitive scholarship. So something that really increases your chances is having a paper or a manuscript published or submitted. And one of the projects that I had been working on the past summer before I applied was wrapping up and was almost ready for publication. But we were running really short on time. I think we had 1 or 2 weeks left before the scholarship deadline. So Dr. Chatterjee and the whole lab really came together in order to make sure that the last couple of experiments or the last bits of writing really got done in order to reach this milestone for me. So that was really amazing to be a part of.
I think that you have even referred to her as your lab mom.
Yes, I have.
And you found her sort of serendipitously because I'm going to kind of paraphrase. You're part of the Honors College at UVM, and through that, you needed to work with a lab or interview a scientist, and that's in part how you found the lab in the first place. Now you're going into your senior year, right? Okay. So what do you love most about being part of the lab?
Yeah, I'd say I love that every single day is different. I love that new research is constantly being published. I'm a very curious person, so I love learning. I love being able to kind of go into lab and learn something new every single day. I love that when I encounter something that interests me, I'm kind of able to talk to my other lab members or Dr. Chatterjee about it. And then if we decide to move forward, we actually have the materials right then and there to be able to test it. So that kind of quest for new knowledge is super rewarding to me. I also think that something that people don't necessarily associate with research is that I kind of view it as a creative process. From the initial design of the experiment to the execution to then being able to kind of communicate that research to others. Everyone's process looks different, so it's kind of allowed me to channel my creativity in a way that I didn't necessarily think I would be able to do in a more sciencey kind of degree.
So what comes after graduation?
Yeah, I'll be applying for PhD programs in the fall. I'm hoping to stay.
I think you're going to be a bit of a get.
I hope so. But yeah, I'm hoping to stay in the cancer biology or the molecular biology field. And then after my PhD, I'm hoping to become a principal investigator. I really like the idea of being able to run my own lab and being able to mentor other students and kind of teaching a class or two on the side. I think it's something that kind of combines all of my interests, and I'm really looking forward to.
Okay, well, your opportunity to mentor students begins right now. What would you like to say to young people in Vermont who aren't sure what their futures hold?
I'd say definitely don't be afraid to take risks and put yourself out there. Don't let the fear of failure kind of hold you back. You need to experience a lot of different things in order to find that one passion that you're super in love with and that you think you can envision yourself doing every single day.
Yeah, and that even if you are, like I know you said that you were a very artistic person and a very creative person, and that so don't rule sciences even out. Like go take all the classes, find what you love. College can sometimes just be that amazing opportunity to discover a talent, a calling that you didn't even know you had.
Yeah.
Well, Madison Guyette, thank you so much for joining me.
Yeah, thank you.
And I'm wishing you all the best. I can't wait for you to treat my cancer someday.
Thank you.
And as always, thank you, Vermont, for learning and exploring with me today. And thanks to the team that makes Across the Fence possible. I'm Amy Finley. This is Across the Fence. Come back and let's learn and explore again tomorrow.